Welcome back to the Skin Physiology and Biochemistry playlist. In this video, we're going to go over the synthesis and biochemistry of some of the monomers of melanin. So we have this big idea that we're going to synthesize a molecule known as melanin. So we're going to be synthesizing melanin, and just to give you some a broad understanding of what melanin is for, melanin is in your skin. It's synthesized by cells called melanocytes, okay, thus the name melanocytes, melanin, okay? And melanin is biosynthesized in these organelles inside the melanocyte known as melanosomes, okay? And the purpose of melanin is to protect the underlying DNA from mutation. Okay, so we have this big idea that UV light comes from the sun. That's a really common wavelength of light that comes from the sun. And UV light, as we saw in previous videos, can cause mutation to DNAs, particularly pyrimidines. You can get things like cyclobutane thymine dimers and so forth, and those things can lead to cancer, and ultimately they can be lethal, ending in death. So you would like a way to protect underlying DNA in the tissues beneath the skin, so melanin is what accomplishes that. But in order to make the polymer known as melanin, we're going to first have to make the monomers of melanin. And in this particular video, I'm neglecting pheomelanin. We're just simply talking about eumelanin, which is this guy right here, eumelanin. Eumelanin is responsible for the blackish, brownish tint to skin. So people of African descent probably are going to have more eumelanin than somebody who's from Scandinavia or something like that. Okay, so we're going to look at the synthesis of the individual monomers, and the monomers basically are going to be these two molecules right here, 5,6-dihydroxyindole, and then we're also going to have a molecule known as 5,6-dihydroxyindole carboxylic acid. And actually, before we go into any real detail on this, I'm going to show you the abbreviations that people typically use to denote these. This one's typically just referred to as DHI for dihydroxyindole. This one's referred to as DHICA, dihydroxyindole carboxylic acid. Okay, so if I abbreviate those as that, you'll know what they mean. Okay. Now, if you're watching this in the context of my anatomy and physiology class, the following three pictures are in your slide. So this picture's in your slide. I'm going to tell you what you need to know from it. This picture's in your slide, and then so is this right here. So the following three pictures are there. This one's sort of new that I found on Google, and I think it's a really good picture to illustrate where everything comes from. Now, what I want to mention at this point is that somehow we're going to have to start this synthesis with an amino acid known as tyrosine. Okay. Now, here's the thing about tyrosine is you can either start this synthesis of these monomers with tyrosine, or you can actually start them with another amino acid that's related called phenylalanine. Okay, phenylalanine is also an amino acid that's found in proteins, and it turns out that there's a reaction known as phenylalanine hydroxylase that can actually convert phenylalanine to tyrosine. Okay, now this particular enzyme, phenylalanine hydroxylase, this is a cytosolic enzyme. It's expressed in the cytosol of the melanocytes. Okay, so in the cytosol, this enzyme gets upregulated when you want to synthesize melanin, and phenylalanine will therefore get converted to tyrosine by phenylalanine hydroxylase. Now, the question is, I mentioned that melanin monomer synthesis and the polymerization right here, this all occurs inside the melanosome, which is an organelle inside the melanocyte, kind of like a lysosome or a peroxisome, although it's different in function. It's just an organelle inside the melanocyte. Okay, but the question is, you have this tyrosine that's in the cytosol of the melanocyte. How do you get it into the melanosome? So let me sort of draw kind of a rough picture of this. So this right here, this is basically your melanocyte. And the melanocyte has these things called dendrites that stick off of it, although those aren't really important here. And you have these organelles called melanosomes. Okay, so this right here, this is a melanosome, and the cell as a whole is a melanocyte. Okay, so the question is, how do I get the amino acid tyrosine? And by the way, tyrosine looks like this when you're looking at it at physiological pH. Just give me 1 second. So this is tyrosine. And the question is, how do you get this guy into the melanosome? Well, it turns out that there are special transporters in the membrane of the melanosome. And these melanosome tyrosine transporters are specific for tyrosine. And so what's able to happen is this tyrosine is able to be transported into the lumen of the melanosome. And that's how the tyrosine gets in there. So this tyrosine right here, this molecule right here, this is of course L-tyrosine. This is essentially pumped into the melanosome through a tyrosine transporter, which I denoted in purple. And then, of course, in the cytosol of the melanocyte, we have phenylalanine, right? And phenylalanine is converted to the tyrosine by phenylalanine hydroxylase, this particular enzyme right here. Okay? So now what we have is we have this tyrosine that's inside the melanosome. Okay. Now, if you go to the other videos on melanocyte function, we go into some of the biosignaling. But what I want to go over very quickly is that there are several enzymes that get upregulated in the melanocyte by presence of a hormone called alpha melanocyte stimulating hormone. And of course, in certain ethnicities of people, you have different levels of activity in the melanocyte. But the main two enzymes we're concerned with in melanin synthesis are these two that I'm going to circle in a vibrant color, light blue. These enzymes are called tyrosinase, arguably the most important of both of these, and then also this enzyme called dopachrome tautomerase, and sometimes you'll hear it referred to as dopachrome isomerase. Now, what I'm going to do in these pictures is I'm going to show you what I feel are the most important things to know. Okay? And in other videos, we'll go into the mechanisms of these enzymes, although that's not directly required for my anatomy and physiology course. Okay? What I want you to know is that there's an enzyme located in the membrane of the melanosome. Okay? So, in fact, what I'll show is that I'm going to sort of kind of blow up this melanosome. Okay, that organelle inside the melanocyte. Here's the melanosome. Okay, and we'll look at this in more detail in other videos. So in here, this is the lumen of the melanosome. And embedded in the membrane of the melanosome, you have the enzyme tyrosinase. Tyrosinase, that's this enzyme up here that's going to convert tyrosine into dopa and do some other reactions as well. And then the other enzyme that we're concerned with that also exists in the membrane is called dopachrome tautomerase, or I'll abbreviate that as DT, dopachrome tautomerase. And in fact, all these reactions that we're going to see in melanin monomer synthesis, they're all occurring here inside the lumen. So that means that of these two enzymes, the catalytically active component of the enzyme is this luminal side of the enzyme. That means the part inside the membrane and outside of it are really just for structure. Okay, but these two enzymes exist in the membrane of the melanosome, and the first one, tyrosinase or tyrosinase, converts tyrosine into dopa. Okay, now dopa also reacts with tyrosinase, and it's going to form a molecule known as dopaquinone. I'm going to box this because it's really important. This is called dopaquinone. And what I'll mention at this point, and I won't go any further on the right side of the pathway in this particular video, but dopaquinone is a branch point. Okay, because what dopaquinone can do is it can go into this pathway on the right, which notice is for pheomelanin. Let me get my brush. This pathway over here is for pheomelanin, or it can go towards the left and go towards the eumelanins. Okay, in which case each monomer of the different pathways is different. Okay. I want to make sure you understand that dopaquinone is a branch point to go to either pheomelanin and eumelanin. Okay? And to better understand that, I want to look at this picture as well over here on the right. Okay? So what we're showing here is dopaquinone right here. So this molecule right here, this is dopaquinone. Okay? And you'll notice that dopaquinone is also converted to dopachrome by this enzyme tyrosinase, as denoted by the key. So tyrosinase both converts dopa to dopaquinone, and then it converts dopaquinone into dopachrome. Okay, so if you're considering eumelanin synthesis, the main branch point I'm concerned about is called dopachrome. Okay, and if you go back over here to this picture right here, you'll see dopachrome can either go and turn into DHI, or it can react with dopachrome tautomerase to form DHICA. Okay? And so basically on this picture what they're showing is that you can take a spontaneous route, meaning a non-enzymatic route. So this route going to the right here is non-enzymatic, and you can end up forming this molecule called 5,6-dihydroxyindole, and this molecule is also abbreviated DHI. Okay? Also, notice B stands for dopachrome tautomerase. So let me do that in pink. So this is dopachrome tautomerase. Dopachrome tautomerase turns dopachrome into this molecule down here, this molecule, which is dihydroxyindole carboxylate, which is also abbreviated DHICA. Okay? And these two molecules right here, these are what we call pre-monomers. Okay, these are the pre-monomers, and they're pre-monomers because they have to go through one more enzymatic step to become the active monomers of melanin. So notice what ends up happening is we're at this sort of point where we have DHI right here, dihydroxyindole. We also have dihydroxyindole carboxylic acid, or DHICA. Now, also what we can show, and let me kind of copy and paste this. So this enzyme right here is tyrosinase. Let me copy and paste that. Tyrosinase has other activities as well. What it can actually do is it can oxidize dihydroxyindole into indole 5,6-quinone, and it can oxidize 5,6-dihydroxyindole carboxylic acid into indole 5,6-quinone carboxylic acid. Let me show you that reaction very quickly. So that's what this reaction is right here. So basically, this is going to be an oxygen-dependent reaction, and actually because it's an oxygen-dependent reaction, sometimes tyrosinase in this context is also called DHI monooxygenase and DHICA monooxygenase. But the whole idea here is, notice what we're doing. We're taking a diphenyl ring, which is this right here, and we're oxidizing it into a quinone type of ring. Okay, and so this enzyme that does this A, this is of course tyrosinase. So you can see that tyrosinase has lots of activities here in melanin synthesis, particularly in eumelanin synthesis. And these two molecules right here, let me box these. These two molecules right here, so we have indole quinone and indole quinone carboxylate. Okay, these two molecules right here, these are what we call the active. These are the active monomers. Okay. These are the active monomers, and then these get polymerized into melanin. These are the active molecules that get polymerized into melanin. And all of these processes that are taking place, these all occur in the melanosome, with the exception of phenylalanine hydroxylase. That occurs in the cytosol of the melanocyte. Okay? So in terms of eumelanin synthesis, let me tell you what I want you to know and to be able to reproduce on the exam. Okay? I want you to be able to tell me the names of the molecules and the enzymes, okay? And let me specifically tell you which names I want you to know, okay? I want you to know that in the cytosol of the melanocyte, phenylalanine is converted to tyrosine, okay? I also want you to know that tyrosine, like I mentioned down here, I want you to know that tyrosine gets transported into the melanosome. Okay, I want you to know that. I want you to know that there's a transporter that moves tyrosine from the cytosol of the melanocyte into the melanosome, the organelle where melanin synthesis takes place. I then want you to know that the rest of the enzymes, tyrosinase and dopachrome tautomerase, are the two main enzymes in melanin synthesis, and they exist in the membrane of the melanosome, as shown right here. Okay, and they catalyze inside the lumen of the melanosome. So I want you to know that. Okay, so now the processes that are taking place inside the melanosome. I want you to know that tyrosinase has multiple functions. Okay, I want you to know that tyrosinase converts tyrosine to dopa. I want you to know that tyrosinase converts dopa to dopaquinone, and then I also want you to know that tyrosinase converts dopaquinone into dopachrome. And I do want you to know that dopachrome is really the branch point in eumelanin synthesis. Okay? I want you to know that dopachrome is the branch point in eumelanin synthesis. Okay? Then what I want you to know is I want you to know that dopachrome reacts in two different pathways, okay? I want you to know that in a spontaneous reaction, meaning that this particular reaction, let me find my mouse, is this is non-enzymatic. I want you to know that in a non-enzymatic reaction, dopachrome converts to DHI, and you're welcome to abbreviate it as DHI, although I would like you to know it's dihydroxyindole. You're welcome to abbreviate it. I want you to know that in a non-enzymatic reaction, dopachrome is converted to DHI. I want you to also know that in a reaction catalyzed by dopachrome tautomerase, I want you to know that dopachrome can be converted to dihydroxyindole carboxylic acid, which you may abbreviate as DHICA. Okay? That being said, I want you to know that those two molecules, these guys right here, so this one, find my mouse, it's hard to find on a white screen. So I want you to know that this molecule right here and this one, these are the pre-monomers. These are the pre-monomers that I mentioned. They're not totally active. They first have to be oxidized by tyrosinase once again. So they have to be oxidized by tyrosinase into their respective quinones. And I do want you to know the names of these quinones. They're indolquinone and indolquinone carboxylic acid. And then I want you to know that from here, the polymerization occurs. Polymerization, and it occurs in the lumen of the melanosome. Okay, and when that polymerization occurs, that's what gets you eumelanin, our final product that we're going to put around the DNA of your skin cells. Okay, specifically outside of the nucleus. Okay, so let's do a quick recap of everything that's happened. So phenylalanine gets converted to tyrosine by phenylalanine hydroxylase in the cytosol. Tyrosinase converts tyrosine to dopa. Tyrosinase converts dopa to dopaquinone. Tyrosinase converts dopaquinone to dopachrome, and dopachrome is the main branch point. In a non-enzymatic reaction, dopachrome can be converted to 5,6-dihydroxyindole, DHI, or in a reaction catalyzed by dopachrome tautomerase, dopachrome can be converted to dihydroxyindole carboxylic acid. Okay? Both of these pre-monomers can be oxidized into their active monomeric forms by tyrosinase, in which DHI gets converted to indolquinone and DHICA gets converted into indolquinone carboxylic acid, and then those guys get polymerized into melanin. Now, one thing I do want to mention, because it is of some importance, is that the more DHI you have, the blacker the tint of the skin will be. Okay, so in other words, the darker it'll be. If you have more of the DHICA, okay, if you have more of that related monomer inside your melanin, then it's more of a brownish tint, meaning it's a little bit lighter. So basically, the ratio of DHI and DHICA inside your melanin actually, in large part, dictates the tint of how black or brown your skin is. So the more DHI there is, the blacker it is. The more DHICA is, the browner it is. Okay, so hopefully that makes a little bit of sense. Okay, now, here's a question that's on your essays for the test. Okay, the question is, what is arguably the most important enzyme in melanin synthesis? Okay, and first of all, your answer should be tyrosinase. And the question is why? Why is it the most important enzyme? Well, notice what it's doing. Number one, it's catalyzing this committed step right here in melanin synthesis by converting tyrosine to dopa. It also converts dopa to dopaquinone, and it does this as well, dopaquinone to dopachrome. And if that wasn't enough, it's also catalyzing the conversion of the pre-monomers into their active monomeric forms. So in other words, what we're saying is that if you have a deficiency of tyrosinase, you won't be able to synthesize eumelanin. Okay? And that actually happens. We'll talk about that in a minute, the implications of that. But also, notice what's happening is you have to have dopaquinone, this guy. You have to have dopaquinone to make pheomelanin as well, right? Because tyrosinase converts dopa to dopaquinone. So if you don't have dopaquinone, this pathway on the right that we haven't talked about yet for pheomelanin, this will also not happen. Okay, because you have to have dopaquinone to make Eumelanin. Specifically, you have to have it to make alanine hydroxybenzothiazine, the monomer of Eumelanin. But if you don't have dopaquinone, you can't make that monomer of Eumelanin, so Eumelanin won't form. Okay, so tyrosinase is important for both eumelanin and Eumelanin synthesis. Now, I want to ask you a question, and this is also in your PowerPoints. What would happen if you had a deficiency of tyrosinase? Okay, and the answer lies over here to the right in the fourth picture. Okay, the answer lies in the fact that if you have a deficiency of tyrosinase, what you'll end up getting is something called albinism. Okay, and albinism is biochemically it's a deficiency of tyrosinase. And that's something very important to realize. If you ever get into the medical field, that's something they'll expect you to know. And this guy that's shown right here, this guy is actually of African descent. Okay, he may not appear like that on the surface, but he's of African descent. But this guy has a deficiency of tyrosinase, and so he's unable to make eumelanin specifically. We're not really concerned about the pheomelanin here. But normally people of African descent, they have lots and lots of eumelanin. Okay, but the problem is that when you have a deficiency of tyrosinase, all those processes that we talked about that tyrosinase catalyzes, they can't happen, and so you'll have a lot of problems making eumelanin, and it'll virtually stop. Okay, so this guy can't make eumelanin, and so he has skin that appears the way sort of a Scandinavian would appear. Okay, but he is of African descent, he just can't make eumelanin. Okay, so that's sort of a medical application of this, and it actually does happen quite a bit. Okay. So hopefully this video gave you a little bit of intuition on eumelanin synthesis. In the next video, we're going to look at the short pathway of going from dopaquinone to a pheomelanin polymer. See you in the next video.